Hi, so for BIO120 this semester, thinking about the projects that you did, there were really four. You did the termite experiment, you did the hydroponics experiment, you did the antibiotic-resistant microbes in soil, and you looked at yeast in response to UV damage and protectants that could possibly protect them against that damage. Now, in preparing for the final, when you're going through the projects in your mind, what I recommend you think about is what was the main purpose or overall question for each of these projects? How did you address that question through your experimentation? So what were your independent, dependent variables? What were your controls? You didn't always have them, but did you have a positive control? Did you have a negative control? What were they? What kind of controlled variables did you have? And what was the overall techniques that you used for that experiment? Then you also need to consider what data did you collect and how did you analyze and interpret that data. Now, I don't want to have this video be too long, so I'll just quickly go through this and then you can follow up with any questions with me later. So for the termites, your overall purpose might have been, why do termites follow this blue line? And you might have had a specific hypothesis of it's the color blue that they're attracted to, right? Your experimental approach might have been something where your independent variable was the color ink that you used to draw a line, and your dependent variable may have been measuring how long a termite placed on a line would remain within, say, 1 centimeter of that line. For controls, you might have had a positive control where you use the blue pen the termites are attracted to, and perhaps no line or a line drawn with a pencil as a negative control. For analyzing the data, you would then do many replicates, so do your experiment repeatedly with several termites and get an average sort of time spent on a red line versus a blue line to see if there was a difference. The hydroponics experiment, the big picture overall purpose question was how can we best support growth of plants using hydroponics media, and how can we make it most cost efficient? So your experimental approach, your independent variable would be the type of media or the combination of media that you use to grow your plants in. Your dependent variable would be plant growth based on weight. Controls, we didn't really have necessarily controls, so your group might not have had a control. Potentially you used controls where you had one quadrant that was entirely rock wool and another quadrant that was just paper towels, one that you thought would well support growth, one that you thought would not support growth well, but not everybody had those controls, and that's okay. For techniques, remember that you had to use the scale for that experiment, and unfortunately we didn't get to do the water holding capacity and air-filled porosity, so we would not test you on those techniques since you didn't actually do them, but we would expect you to know how to use the scale. For data analyses, essentially you just weighed your plants at the end and subtracted out the cost of the materials to produce that plant. The soil microbe experiment, the overall purpose question was to see are there patterns in which soil areas have high percentages versus low percentages of antibiotic resistant microbes. So your experimental approach, your independent variable was where you collected your soil sample from. Your dependent variable were the number of colonies on plates with and without antibiotic, to therefore measure actually what percentage of microbes in your sample were resistant to the antibiotic. For controls, you had plates with no antibiotic to know how many microbes were in that original soil sample altogether. For techniques, you needed to go over pipetting, serial dilutions, sterile technique, plating, and counting microbes, colonies. For data analyses, you essentially had to be able to take colony counts and translate that back to how many cells did you originally have in your gram of soil. And also from those counts based on all cells compared to antibiotic-resistant bacteria, you should have been able to calculate a percentage of resistant bacteria. Your yeast projects were broken down into a baseline experiment and a protectant experiment. For the baseline, the overall purpose was to see how yeast respond to UV damage, how lethal is this UV to them, and how important is the ability of cells to repair their DNA in response to DNA damage. How much does that help increase their survival? For your protectant experiments, the overall purpose would obviously be, can your potential protectant, does it actually protect against UV damage? Your experimental approach, independent variable, would be, for the baseline experiment, amount of UV exposure. For the protectant experiment, it would be with and without protectant, and your dependent variable in both cases are colony counts or survival. Controls, you always had plates that were not exposed to any UV to get maximum survival, and then you always had plates that were exposed to a high level of UV for your baseline experiment, or to UV without any protectant for your protectant experiment for a low level of expected survival. Techniques, again, you used the micropipettes, you used sterile technique, you used plating, and you used colony counting. Data analyses here, we really worked on how to standardize data, and again, how to combine replicates to get stronger results.